Okay, so it's my pleasure to open this international meeting about skin physiology. I will host the first session about skin homeostasis, and we will have 3 speakers, one from Uppsala, Sweden, one from Chicago, US, and one from Amsterdam, Netherlands. And I'm happy to be here in Vichy. Vichy is a known city for spring waters, known like Spa in Belgium or Baden-Baden in Germany or Bath in UK, and we have a member of Bath here. I will welcome Eniko SONKOLY. Eniko SONKOLY is a professor of dermatology at the Department of Medical Science in Uppsala. She earned a MD degree from Semmelweis University in Budapest in Hungary in 2002, and a PhD degree in immunology at the Department of Dermatology of University of Szeged. In 2005, and she do also a postdoctoral research training in the Department of Heinrich Heine University in Düsseldorf, Germany, and then she moved to Karolinska Institutet in Stockholm, Sweden in 2006, and finally to Uppsala University in 2022. So her research focuses on the molecular and immunological background of psoriasis and atopic dermatitis, particularly on the roles of non-coding regulatory RNAs, such microRNAs and long non-coding RNAs in chronic skin inflammation. But she's also the new president of the ESDR after Elie Sprecher. And the most important is that I discover after some research that she's the wife of Andy Pivarski, who was a speaker in this meeting 12 years ago. So you are like at home here. So we will hear you about non-coding RNAs in skin health and diseases. Please.
Thank you very much. Yes, thank you very much for this nice introduction, and I would like to start with thanking the organizers for inviting me here. It is a pleasure to be in Vichy, second time, as you mentioned, and I'm really sorry that we brought some cord and snow. I hope it's not our fault, from Sweden. It is also a great pleasure for me to talk to you about non-coding RNAs in skin health and disease. So all of you know skin. This is the topic of this meeting. But if we think about what is skin made of, it is made of, to a large extent, just like our other organs, of proteins. But what defines how these proteins are expressed? Which proteins are there, in which cells, and at what time? This is very complex. And I would like to go back here to the information flow, the central dogma of molecular biology, which is from the 50s, which says that DNA makes RNA, makes proteins. So all the information that is needed to make the proteins is encoded in our DNA, which is in the cell nucleus, and through this process of transcription, which results in messenger RNAs, which are just intermediates, and then translation, we get the proteins. And this makes skin. So DNA makes RNA, makes protein, makes skin. Very simple, right? But there is one thing that is a little bit disturbing, maybe, because the human genome contains 20,000 genes, 20,000 genes, and that is actually not more than the tiny little worms. And we can think about, like, well, maybe we are a little bit more complex than the tiny little worms, maybe. And then the difference is not in the number of protein-coding genes. So where is the difference then? So the surprising discovery was, after the ENCODE and PHANTOM projects, that actually most of our DNA does not code for protein. It's only 2%. But most of it is actually transcribed to RNA. And this was first regarded as something as junk. But if we think about going back to the worms and the question of complexity, the percentage of non-coding DNA actually increases with increasing complexity, not like the number of protein-coding genes. So there might be something important there. Moreover, if we look at the RNA, which is not coding for proteins, which we call today non-coding RNA, the percentage of non-coding RNA also increases with complexity. So the more complex the organism, the higher the number of non-coding RNAs. And that tells us that it could probably do something. And it was first thought that it is junk, but then it turned out probably not all of them, at least, are junk. So if we look at it this way, actually, humans, we have more non-coding than protein-coding genes. So if this is the genome, this is the part which is the transcriptome, which is transcribed, and this is the small part which makes protein. And many of these RNAs, today we have learned a lot about them, and many of these RNAs actually have a function. So we have to redefine the concept of a gene. So a gene is not synonymous to protein-coding genes, which also revised the central dogma of molecular biology, which is DNA makes RNA, which sometimes does not code for a protein, but functions as RNA. So how could they function as RNA? They regulate other genes. And this I show you with the example of microRNAs. And microRNAs, they are so small they cannot even code for a protein, but they work as RNA, and they regulate the expression of other genes. And they were actually coming back to the worms. I know I always come back to this, but we come back to the worms. They were discovered in the small worm, Caenorhabditis elegans, the elegant small worm, and it was thought that this mechanism is something that is a peculiarity in worms in the 90s when it was discovered. But today, it turned out that this is a fundamental mechanism of gene regulation, and it is so important that even the Nobel Committee this year thought it was very important, and so the Nobel Prize was awarded to the discoverers of this mechanism, Victor Ambros and Gary Ruvkun, this year, 2024. So they will receive their prize this December for the discovery of this fundamental mechanism. So what are these microRNAs? They are tiny RNAs, but they have a profound physiological importance. They are very short, approximately 22 nucleotides, and they regulate this process, which I mentioned in the beginning, which we call gene expression from DNA to RNA to protein, and they regulate here. And actually, most of our protein-coding genes are regulated by microRNAs, approximately 60%. So if you have your favorite protein-coding gene, the chances that the microRNA regulates it are higher than that it is not regulated by microRNAs. And accordingly, these microRNAs are involved in all biological processes where they have been studied. It would be very simple if one microRNA would regulate just one protein-coding gene, then it would be actually a silencing RNA. It's rather like that, since they regulate their targets through a base pairing, which is not complete. So they have several targets, dozens to hundreds of targets, which they regulate even at the same time and in the same cell type. And by this mechanism, they can regulate whole gene networks. Very often, their targets can be in the same regulatory pathway, and that makes them very powerful regulators. Of course, it doesn't make our understanding of gene regulation more simple. It's rather a bit even more complex. So what does it all have to do with skin, you may ask? We are at the skin meeting. So microRNAs in skin, what do we know about this? I will first give you a short historical overview, and then I will show you some of our results from an ongoing project. So if we go back in time, 2006, it was discovered that microRNAs are actually essential for skin development. So if you delete DICER1, which is a gene encoding for a protein which is needed for the processing of microRNAs. What happens then? In these mice, they die a few days after birth because of dehydration. They also have defective hair growth. They have a decreased number of granules in the granular layer and so on. So they have such abnormalities in skin that it turns out that microRNAs are very important for skin development. And if you delete another enzyme which is essential for microRNA processing, you get the same phenotype. So this was the start. At that time, we were thinking about, is it possible that microRNAs could contribute to skin diseases? Since they are so important in regulating biological processes and also in skin development, is it possible they could contribute to skin diseases? Nothing was known about it at that time. So now we are back in 2006-07. We were looking into the role of microRNAs in inflammatory skin diseases, psoriasis and atopic dermatitis. These are the two most common chronic inflammatory skin diseases in humans. And we compared then their microRNA expression patterns, and we saw that psoriasis and atopic dermatitis have actually very specific microRNA expression patterns in the skin. So they were dysregulated, many of these microRNAs, and this was something completely new at the time. And the microRNAs that we identified, many of them we have actually studied their function afterwards. I will show you one example. When we looked at these microRNAs closer, we wanted to know, like, where are they expressed, and nothing was known about that. And then we saw that among healthy organs, miR-203 was one of the microRNAs which we found very interesting because it was very specifically expressed in skin, and within the skin, it was specifically expressed in keratinocytes. So we thought that this might have an important role in the function of keratinocytes in skin homeostasis. And then when we looked at its expression pattern, then we saw that it was expressed in the differentiated keratinocytes in the upper layers of the epidermis, but not in the basal layers. So our hypothesis was that it could regulate the differentiation process of the keratinocytes. And then we decided to follow this up, and we were working on this microRNA when this came out. This very important paper from the Fuchs group in Nature in 2008, and then they showed that miR-203 is very important, actually, and it promotes differentiation by repressing stemness. And you can see the expression pattern in mouse skin, which is somewhat similar to what we saw in the human skin. So it turned out that this is actually a very important microRNA, and they termed it as a skin microRNA. And it is also very evolutionarily conserved, so even the fish have it. So after that, we worked on this microRNA for many years, and also others worked on it, as you can see, and all of this research led to the conclusion that miR-203 is actually a very important skin microRNA, and it governs the differentiation process of the keratinocytes. Under normal conditions, miR-203 gets expressed in the differentiated keratinocytes, and through stabilization of this regulatory circle, it allows differentiation, which is a one-way road. The keratinocytes do not turn back. However, in cancer, this circuit is stabilized in another way, in which miR-203 is kept low, and these cells then do not differentiate, but they proliferate more, and this is what happens, for example, in basal cell carcinoma or squamous cell carcinoma. And we did all these studies together with Andor Pivaci, and we really characterized the function of this microRNA, and many other groups did that also. So now we are convinced that this is one important example, but is this the only one? And the answer is no. This is not the only one. This is a very important one, but there are also other microRNAs in the skin which regulate important cellular functions in keratinocytes or in immune cells. These are some of the examples. This list is absolutely not complete. So if you are missing something, then it's because this is not complete. This is just to show you some examples of microRNAs which we have studied throughout years and found that they regulate important cellular functions. And by this, they can contribute to the pathogenesis of inflammatory skin diseases, for example, by making keratinocytes more responsive to inflammatory cytokines. So since then, microRNAs have been studied in many different aspects of skin health and disease. MicroRNAs have been proven to be important in wound healing. Several microRNAs they are expressed dynamically through the different phases of wound healing, and they can regulate inflammation, angiogenesis, and reepithelialization, and so on, which was studied, for example, by Ningsu Landén and her team at Karolinska Institute, but also by many others. During aging, it turned out that microRNAs are very important there as well, and that aging actually has a robust impact on the microRNA expression in the skin. This is one project which Andor Pivarczy's group has done, and we have collaborated on this. We looked at the microRNA expression in young and old skin, and we have found that the microRNA expression is changing during aging in the skin. These were full depth skin biopsies, and especially photoaging, but also during chronological aging. And here I would also like to mention the work of Jérôme Lamartine's group, who is in the organizing committee here. He has studied the effect of aging on microRNAs in epidermal keratinocytes, and so that microRNA expression specifically in keratinocytes changes during aging, and this is one example for that the MIR130, whose expression is increasing during aging, and it turned out that it regulates epidermal differentiation. Moreover, microRNAs are also in extracellular vesicles, so the cells can secrete them, and this is also something that is changing, a process which is changing during the aging. Now, of course, these processes like inflammation, wound healing, which I mentioned before, they also have to do with immune functions of the keratinocytes. This is something I would like to mention here also, that microRNAs are involved in the innate immune functions of the keratinocytes. For example, if we treat keratinocytes with ligands of toll-like receptors, then many microRNAs, or some of the microRNAs can change their expression. There are some which seem to be important in this process, for example, MIR146A, which is regulated by multiple toll-like receptor ligands. And today we know also that some of these microRNAs are important in regulating the response of keratinocytes to toll-like receptor ligands. But here I would also like to say that there are still many things that we don't know. So we know that there are microRNAs that regulate this process, but I think that there is a lot that is still unexplored. And then, in the past years, we wanted to know like if microRNAs have a role in keratinocytes, in skin inflammation. So in our previous studies, we looked at the full-depth skin biopsies. And as you know, skin is composed of many different cell types. So if we look at the microRNA expression pattern in the full-depth skin, then it's like we are looking at a smoothie, because we take all the skin cell types, we mix it, we homogenize it, we make our smoothie, and then we compare, healthy skin and psoriasis. This is how it looks like. And then we get an average of expression. But we wanted to know a little bit more in detail. We wanted to look at the dragon fruit. And then if you want to look at the dragon fruit, which represents the keratinocytes here, then you don't want to make a smoothie, right? And to solve this problem, we have had different approaches. So we took punch biopsies from psoriasis skin and from healthy skin, And we took the epidermis, and then from the epidermis we made single-cell suspensions, and then from there we took different approaches. One of them was that we removed the CD45 positive cells, the immune cells, so then we have mostly the keratinocytes left. And from this cell population, we characterized the messenger RNA, the microRNA, and long-encoding RNA expression, and in another approach, we also did single-cell RNA sequencing in the epidermis. But here, I would only like to focus on the microRNAs, because from this screen, we found one microRNA which we found extremely interesting and which is something that we are working on at the moment. So miR-149 was one of the microRNAs that was suppressed in the epidermis of psoriasis skin as compared to healthy skin. And if you look at this in situ hybridization, for example, it looks like it's almost completely lost. And we wanted to know what happens here, what is the reason for it, and what are the consequences for it. And we mimicked the loss of microRNA by inhibiting miR-149 in cultured keratinocytes using a specific inhibitor. And then we looked at gene expression, what happens then? And what we found was that if we inhibit miR-149, then actually a lot of inflammatory genes get induced, even without any inflammatory trigger. So you can see IL-6 and TNF and IL-1 and so on. So we got an inflammatory profile. And this is something that we thought it was very interesting because it said us that the suppression of miR-149 could contribute to the psoriatic phenotype, which is an inflammatory phenotype. And long story short, after many years of research, this is the model that we built, that miR-149 is suppressed in the psoriatic keratinocytes by interferon gamma signaling, and its suppression, it results in the upregulation of TWEAK receptor, which is the direct target of miR-149. I showed you microRNA targeting before. This turned out to be a direct target. And the TWEAK receptor is the receptor of TWEAK, which is a TNF superfamily member, an inflammatory cytokine, which had been associated with psoriasis, but also atopic dermatitis. So increased expression of TWEAK receptor leads to an induction of inflammatory signaling. So this was the model that we built from the results of our experiments, but we wanted to know more. We wanted to know what is the role of this microRNA in skin physiology? And to dig deeper into it, we generated mice with epidermis-specific deletion of this microRNA. And then what happened? What happened was that we generated these mice and they were completely normal. So we were a little bit disappointed. There was absolutely no phenotype. We looked at the skin, the hair, the nails, the mouth, and so on, the teeth, everything was normal. We let them age, they were still normal. We looked at in the microscope, it was still normal. So the story could have ended here, maybe. But then we were also looking at the molecular level, and in the transcriptomic analysis is what we found that there was something, because there was a slight inflammatory signature which corresponded to what we saw in the human keratinocytes in vitro, with an upregulation of the direct target of miR-149, the TWEAK receptor, but also some inflammatory chemokines, among others CXCL10. So therefore, we went further. We isolated keratinocytes from these mice, and then we saw that actually, even without any stimulation, they have a higher expression of some of the inflammatory chemokines. But if we treat them with TWEAK, which is then the ligand of the TWEAK receptor, then they are more responsive to that. Okay, but then the mice were still normal, but we wanted to know, okay, but what happens if we challenge them? So what happens under inflammatory conditions? Is it possible that we see any difference there? So for that, we treated the mice with IL-23, and this is a model for psoriasis that is commonly used. We injected recombinant IL-23 into their ears, and then we watched what happens. So what happened was actually that these mice were really more responsive to this challenge. Their ear thickness was higher. The inflammatory infiltrate was much denser. They had also a thicker epidermis. and they had a higher expression of inflammatory mediators. So they had an increased inflammation, psoriasis-like inflammation, which was the consequence of the epidermis-specific deletion of miR-149. And then we wanted to also know that, okay, but we see this inflammatory infiltrate, but what kind of cells are there and why are they there? Because if you remember, we only deleted miR-149 in the keratinocytes. So to find out what cells are there, we also did a single-cell sequencing analysis together with a protein surface marker analysis. And what we could see was actually that we had more of all the immune cells in the knockout animals as compared to the wild-type ones. And especially the myeloid cell compartment and the mast cell compartment was even more expanded. So we could see that if we delete this microRNA, there will be an induced infiltration of both mast cells and myeloid cells, especially them, but also other immune cells. So this is something that is still ongoing, and we are looking into that. But of course, the question that is in I guess, everyone's head now is, okay, well, it's nice, but could we use this to if we would replace this microRNA, would it really suppress inflammation? Can we use it like that? And then, of course, we asked this question, and when we treated the mice with synthetic miR-149, first we induced skin inflammation. This is another model of psoriasis, which is induced by imiquimod. And if we inject synthetic miR-149 into these mice, then we looked what happens. And actually, the synthetic miR-149 could reduce the psoriasis-like skin inflammation. In this mouse model, they had thinner epidermis, and if you look also, they had less redness, thickness, and scaling, and they had less IL-17A, so they had less inflammation. So it really had an effect on skin inflammation. So if we summarize, we think that miR-149 is very important microRNA in epithelial immunity. It should be there under normal conditions, and what happens in psoriasis, it gets suppressed, and then many other inflammatory mediators are induced. And this is, of course, a very short summary, but we think that actually miR-149, this is not specific for psoriasis. This mechanism could be also in other skin diseases and also in other physiological processes. This is something that we are still exploring. And here I would just like to give you a very short teaser that I focused this lecture on microRNAs, but there is also another word which just starting to open for us, and that's the word of long non-coding RNAs. So long non-coding RNAs, which are still RNAs that do not code for protein, they're longer than 200, some say 500 nucleotides. We still know very little about how they exactly work. We know for some of them, but for many of them we don't know. If you look at the expression of long non-coding RNAs, and this is ongoing work, you can see that there are many long non-coding RNAs which are enriched in the skin. So here's the skin compared to other organs. And one of the long non-coding RNAs we have, together with Andor Pivarci, we have ongoing investigations. This is something which we called TIDA. And you can see that it has a very specific expression pattern in the uppermost granular layer of the epidermis. And I don't want to show you more because this could be the topic of another talk. And I would just like to say that, RNAs which do not code for protein, they should not be ignored. There is a hidden layer of non-coding RNAs in the skin. They orchestrate the expression of protein-coding genes. So they are very important in that sense. And microRNAs and also long non-coding RNAs, they can contribute to skin homeostasis and disease, both to aging, chronic wounds, inflammation, and cancer, which I didn't mention in this talk. Of course, the work that I have shown, which is like part of our work, it is a result of teamwork. I would like to thank everybody who contributed to this work, the present and past group members and also our collaborators, and of course, special thanks to Andor Pivarci. We have done most of these investigations together. And last but not least, I would like to, as a president of the ESDR, I would like to invite you all to participate on the 54th annual ESDR meeting, which will be in Antwerp in 2025, the 10th to 13th of September. So I hope to see you all there. Thank you very much.
Really a pleasure to hear you at any time, Eniko. Thank you for everything. So I have two questions. The first one is probably correlated with the work that we have done with Andor a few years ago, but so do you know if some specific strains living on the skin or coming from the gut can regulate, for example, MER 149?
If some specific strains as a microbial,
yeah.
Yes. Actually, we don't know, but I think it would be very interesting to look at. So this is really just the tip of the iceberg. We have looked at the inflammation, but we have not looked into, for example, like the microbial component, or which we would be also very interested in viral infections and so on. But it would be, of course, very interesting to look into that. We have not done that. What I can say is that in the gut, there have been actually knockout mice created by a Chinese group, and they published that that these mice, which lack MER 149, that they have also an increased inflammation in the gut. So that's something that I can say, but they also didn't look at the microbes.
Thank you.
Thank you very much. My question is about the range of action of microRNAs or non-coding RNAs the whole. Is it tissue-specific, or can it act at distance, for example?
So microRNAs are typically thought to work within a cell. So if that is the question, and then they act in the cell in which they are expressed in, but they can also be, which I just mentioned very, very briefly, but they can also be secreted in extracellular vesicles. And these extracellular vesicles can be taken up by other cells, and the microRNAs could have an action. And this is something that I think it has been studied for many years, but I think that there is still a lot that we don't really know, like which cells can take them up, and which microRNAs are in there, and are they really active, and which ones are active. So there are still a lot of open questions.
Okay, last question, Jérôme Lamartine.
Well, thank you, Eniko, for this very nice overview of microRNA in skin. I have a question about microRNA and epidermal stem cells. So you mentioned the work about MER 203 and inhibiting the stemness, but what about microRNA linked to stem cells and being involved in maintenance of stemness in skin? Do you have a mention of sorts of microRNA that have been identified for that?
Yeah, well, actually, MER 203 is this very first and very famous example for that. And they are needed. I mean, this MER 203 is really needed for when it's upregulated, then the cells really leave the stem cell state.
They leave the stem cells, but to maintain the stem cells.
But to maintain them, this is a very good question, and this is something that we ourselves have not worked on. But I think that this is something which should be investigated more for sure. Yeah.
But because it's difficult to identify the stem cells.
This is actually the first very difficult step. Like which cells are really the stem cells? Some call them also progenitor cells, and so that's where the difficulties start. But definitely it's a very interesting and important question.
Thank you. Thank you very much.
Thank you very much, Eniko.
Thank you.